So I'm going to move on to the tutorial. I do have Figini preloaded here. So what I'm going to do is I'm going to actually exit out of here just to sort of show people what happened. So once you click the Jupyter binder link that that I sent out earlier, hopefully people have it working. So you should get a page that looks like this. And what you want to do is you want to click on terminal, and that'll open up the virtual terminal session. Okay, let me just open up my. Okay. All right. So once you're in the terminal, let's take a look around. Let's see where we are. We have. Okay. So basically, you can see the entirety of the GitHub repository here. So actually, a quick note is that you can also copy and paste the commands from a walkthrough that's on the same GitHub page that I sent out earlier. So the walkthrough begins right underneath the launch binder link, right? So this is basically where I'm going to be copying and pasting commands out of, just so if people want to do that as well and not have to type stuff out. Okay, so let's go into the Figini directory and see what that looks like. ls. All right, so we have a number of files here. The first thing I want to do is I want to print the Figini help page and go over some of the options here. There's not a lot of them. Actually, there is a new version that's about to be released that's going to include about 4 more flags that I think are going to be fairly self-explanatory, but I kind of want to go over these ones just because these are the important ones, the required ones. So binder, this is the directory of bins. Actually, this is an archaic name, and I really should change it because this could be a directory of assemblies or genomes or transcriptome assemblies, not just bins. But when I designed this tool for Nancy initially, she had a collection of metagenome assembled genomes, so metagenome bins. And that's what the tool was geared for, and I actually haven't changed the name of this flag since. But that's basically the directory of your genomes or your FASTA files that you provide. The bin extension is the file name extension. So if you have other files in that directory that are not FASTA files, you don't want to include them. You want to provide an extension for all the files that are FASTA files. So ideally, that would be .fasta or FNA or FA. So the -D, so this is actually goes back to the question that was asked earlier. This is the the genomic clustering that's sort of done when you identify genes. At what point do you consider them in the same genomic neighborhood? So if you have 2 genes and they're 5 genes apart, if there's 5 genes that didn't have any hits to any of the HMMs, in between 2 genes that did have hits to some iron genes, would they be considered in the same genomic neighborhood? And you want them clustered. And that sort of goes, that'll be, once you look at the output is sort of organized in a way where you could look at the genome in basically in different neighborhoods. So the different neighborhoods are sort of separated out on the file, so it's easier to inspect. So that's basically what this refers to. So the default is 5, but you could set it to however big you want it to be or however stringent you want it to be. But it'll be more clear once we look at the output exactly what this is doing. The -ref, this is I mentioned this during the lecture. If you want to provide a database for cross-referencing, you could do it using this flag. -out, this is basically the file name. Well, this is the name of the directory that's going to be created, and the directory is going to have all the output files for you. This is really just for, doesn't really affect anything of how Figini runs, only the values that you get on the heat map spreadsheet. So when you run Figini, it'll identify iron genes in your genomes or your assemblies, and in the heat map spreadsheet summary, it will report, by default, it will report the number of genes for each category that you get back. If you want to add the -- norm flag, it's going to normalize the number of genes you get for each assembly to the total number of genes in that assembly or genome. So it'll normalize. The -- inflation flag is something that if you want to multiply that normalized number by. So actually, I've been meaning to set the default to 100. So if the -- inflation is 100, then the values that you'll see in the heat map summary will be percentages. So if you go with the default of 1,000, it's going to be will be percentages. But actually, I've been meaning to change this to 100, which I'll do as soon as this lecture is over. -T is the number of threads that you want Figini to use. Basically, the only parallelizable part of Figini are the HMM search and Diamond Blast. And those are actually the most time-intensive ones. So if you're working off a cluster and you can provide 16 threads, you can do that. Or if you're working off your Mac, then you can provide as many threads as you have available. So these BAM options are actually really cool. This is a recent addition. So like I mentioned, Figini by default reports the total number of genes for each gene category as laid out in the presentation. But you can actually provide a BAM file. So if you're dealing with a metagenome or if you have metatranscriptomes, and you can provide assemblies, or if you have a genome that you've mapped a transcriptome to, you can provide that BAM file. So for those that aren't familiar, BAM files are basically, it's a file that measures coverage, read coverage of a genome or a metagenome. So if you provide the BAM file, when you get the heat map summary file, instead of gene counts for each gene category, you're going to have the values are going to correspond to the read coverage that's calculated from the BAM file. Yeah. So that's easy enough if you have, let's say, hypothetically, if you have one metagenome and you have 20 bins that you've generated from the metagenome, then you map your reads back to those bins, and then you generate a BAM file. You can provide that BAM file. But if you have multiple assemblies, or if you have multiple metatranscriptomes that you map back to your genomes or your metagenomes, you'll have multiple BAM files. And I took that into account, and you can provide those BAM files with this way. So it will be the -BAMs, and then the file that you need to provide this argument is a text file, and I have some sample BAMs files that you can provide here. So let's look into one and see what it looks like. So it's a tab delimited file where the first column is the name of your assembly, and then each subsequent column that's tab delimited is a BAM file that corresponds to each assembly. So as you can see, it's very versatile. You could have any number of combinations of BAM files for assemblies. For example, if you did 3 assemblies and you mapped reads back from each assembly, you'll have BAM files that correspond like this. But let's say you also include a co-assembly where you combine the reads from libraries A, B, and C, and then you mapped all 3 libraries individually to that co-assembly. You'll have 3 BAM files for this co-assembly, and you can provide them like this or like this. If you're dealing with, let's look at another sample BAMs file. So if you're dealing with a directory of genome bins, and you have different libraries for each one, different transcriptomes, for example, you could have something that looks like this, where bins 1 through 7 each have their own libraries, coverage files, but then bin 8, for example, has 2 transcriptomes associated with it, or 2 metagenomes associated with it. So you can provide 2 BAM files like this. Anyway, so this is pretty flexible in terms of how you want how you want PGenie to report the data. If you're providing metagenome assemblies that don't have any gene calls that you've done, and you want PGenie to run protocol for you, you should add the -- meta flag, and that will ensure that protocol runs with the correct parameters for metagenomes. The -- make plots. This will run RScript for you on the heat map file that you created. I'm not going to run make plots here because there's certain permissions errors that I've been getting with Jupyter Binder that I haven't really figured out yet. So we're just going to use the core output of PGenie to look at our results. I found that the -- make plots option works best if you are working with, if you're working off a personal Mac. And that's because some of the R packages that need to be installed using RScript do require super user permissions, and I've had issues where I wasn't able to install all of them on the server that I don't have super user permissions on, and then that usually causes the RScript to crash. But that doesn't really affect the main output of PGenie, which is the CSV files that you get out. Okay, so that's sort of it for the help page, help menu. So why don't we go ahead and, oh, one more thing is that I did include a subsample of the RefSeq database here. It's only 63 megabytes, so this is a very, very small subset of the RefSeq database, and I didn't include the whole thing because I couldn't get the whole thing onto the Jupyter Binder page. So we're just gonna run it with the subsampled database. But first, let's just run PGenie and see what it looks like. So we're gonna start with PGenie bin directory, and then we're going to provide it with the genomes. Actually, let's look at the genomes and see what the genomes look like. So I included 5 genomes, just because not in any particular reason why I included these ones. Just, well, Schuanella is a known iron reducer, and Acidothiobacillus is a known iron oxidizer, and Pseudomonas is a known to produce pyridine siderophores. So, and we have a known magnetosome former here. So that's why I included these actually. So let's start. PGenie bin directory, we're going to provide it with the directory genomes, bin extension. So the file name extension here is .fna, so we're going to include fna here. And we're going to name our output file. I'm just going to call it PGenie out. I'm not feeling very creative, so hit enter. And actually, I ran this before I started the tutorial. So there is already a folder called PGenie out. So if you set a folder that already exists, it's going to ask you whether you want to potentially override it. So we're going to hit yes. And there it goes. Okay. So it's going to take about a minute to run. It's going to take a little bit longer for the siderophores because those are, the siderophore synthesis genes are really long, so those HMMs are huge. So it's going to take a little while, not more than a few minutes. While this is running, are there any questions?
I have a question about the process of making the program. How hard is it to add different flags and functions, and what's your process? I'm just curious.
You mean to add different flags to the actual script, to the actual program?
Yeah, to the actual program. Like, how do you guys decide what's next? And is it a group effort? Or are you just coding?
Yeah, it's a group effort in terms of deciding which flags to add. It's sort of for me, it's basically trying to figure out whether it's computationally possible. It's not really hard to add the flags. but anything any parameter that's involved in Fujini could be added as a flag or as a potential metric. So anything e-value, bit scores, anything related to that, it's really not that hard to add a flag. It's actually I've never actually gotten opposition to adding a flag that I wanted to add to Fujini. Usually I get more suggestions for features to add than I have time to fulfill.
Also, there's a question in the chat box about FNA files. Are they nucleotide or AA files?
Okay. Tanya, the FNA files, are they nucleotides or AA files? It's a good question. So they are actually nucleotide files. As soon as this is over, I'm actually going to look at one of them just to show you what it looks like. But that's a good question. So FNA, as far as I know, according to the NTBI, file name nomenclature, FNA refers to FASTA nucleotide, and FAA is FASTA amino acid. As far as I've seen, FNA generally refers to contigs, and FFN refers to nucleotides that are gene calls, and FAA refers to gene calls that are in amino acid protein sequence format. So the FNA files that I provided here are they're contigs in FNA format. Yeah. So this is okay. So it's almost done. There it goes. So it tells you where the results are. So it wrote one file for visual inspection, and we're going to take a look at this one. And the file for visual inspection has the, well, let's just look at it. Well, actually, let's look at the genome files first. Let's just look at this. Basically, this is it. This is just contigs. So you could also provide amino acid files. And if you provide the amino acid files, you have to add the --orfs flag. I didn't talk about that. But if you provide a directory that has just files in the amino acid format, just the gene files, you can do that. And then you provide the --orfs flag, and then it's going to use those genes. Okay, so let's look at the output. Okay, so we have the heat map data, the gene summary. So the gene summary is, well, okay, I like to look at files with a ~S flag because it doesn't sort of cuts out like this and it's easier to look at. So basically we have a bunch of output. Okay, so I found this hard to look at. So I like to look at the clustered version of the output, which sort of, whoops, let's do the -S version. Okay, so this uses hashtags or pound signs or whatever you want to call these to separate out the different sort of genomic neighborhoods, which have been predefined by the clustering algorithm. So, for example, in acetylthiobacillus froxidans, it clustered XBB and XBD because they're right next to each other on the genome. As you can see, they're encoded. XBB is gene number 458 on this contig, and XBD is gene number 459. Psych1 and Psych2, these are two iron oxidase. Well, Psych2 is a potential iron oxidase. They're also encoded right next to each other, as you can see, 1312, and then Psych1 is gene 1313. Right, so I find that this is sort of easier to interpret or to look at, just sort of peruse the data. Yeah, so this is basically it. So you have the iron gene category here as the first column. These are comma delimited, so if you open this up in Excel, it'll be a lot easier to look at. This is the name of the bin or the genome. This is the gene call ID. Then you have the actual HMM hit. So there's an HMM that's specific to this family, or Psych2, for example. And then the following two columns are basically telling you how significant of a hit that is. And the first number represents the actual bit score. for the HMM hit, and then the second number corresponds to the bit score cutoff for that HMM. So if you have a bit score cutoff of 27.8 and an actual bit score of 333, I'd say that's a pretty good hit. If you have a bit score cutoff of 27.8 and then you look at the bit score and it's 27.9, I'd be a lot more careful with that gene hit. Next thing it looks at is the number of heme binding motifs, and there's one heme binding motif in psych 1, and there's 2 heme binding motifs in psych 2. So that makes sense. And then it provides you with the gene sequence in amino acid format. So then you could blast this yourself. So why don't we let's try this. Let's try another run of Fijini. Actually, let's take a quick look at the heat map output. So the heat map looks like this. Also, a lot easier to read in Excel if you have it. Basically, it's a summary where the different rows are the different gene categories, and each column is for a particular genome that you've provided. Right. So this is by default going to be not normalized, the total number of gene hits for each gene category. Like I said, this is a highly flexible output, and you can really customize it by adding BAM files or normalizing however you want. But in the end, once you run RScript, you'll have a dot plot that sort of represents the data pretty nicely, I think, either way. So let's run Fijini with the let's run Fijini again, but instead of providing with the genomes, we're going to provide it with a different directory. So these are ORF calls. So let's say you already have a set of bins that you've annotated, and you have protein files for them. Look like this. So you have gene calls. So you want to annotate these. So what you want to do is you want to Fijini, and you want to provide this ORF calls directory. We'll see that the file name extension here is .faa. So we're going to do -bin extension FAA. We're going to add the --ORF flag because we're providing open reading frames rather than contigs. And we're going to name our output directory Fijini.orfs. And let's provide a reference data set. Let's provide -RefSeq. So we're going to provide the subsampled RefSeq protein database. Then we're going to enter, and it's going to start running. Okay, so it's going to take another minute to run. It's not going to take very long to do the cross-reference. That's actually the longest step if you want to do the cross-reference. But here, it's just a subsampled RefSeq database. It's about 60 megabytes, so it's not going to take long. It's going to take less than a minute. But if you want to cross-reference against the entire RefSeq database, which is, I believe, 60 gigabytes, then that might take a while. It might take upwards of an hour. Another quick note about the results that we're about to see is that since this is a subsampled RefSeq database, it's not all of the identified iron genes are going to have hits. And that's not going to be for any other reason than that we don't have. It's just chances are that the gene is not in the subsampled RefSeq database because it represents, I believe, 0.1% of the entire RefSeq database. So if there's a certain siderophore synthase that's not says no hit, then that's why. Okay, so it finished analyzing. Now it's looking for the Thurman-Kola S-layer cytochromes and the Geobacter related porn cytochromes. So now it's doing the blast search against the RefSeq database. I didn't include Thurman-Kola S-layer cytochromes because I wasn't able to build HMMs off of those. To build an HMM, I really want to have a good alignment of a set of proteins that are related. And when I went on NCBI with these Thurman-Kola S-layer cytochromes, I couldn't find any good hits. And that's why I wasn't able to make any any HMMs, so I just blast that. Okay, so in this output, basically you have same thing up to so you have the heme binding motif, but then after the heme binding motif, instead of the gene sequence, which you have later on, you have the closest hit in the RefSeq database that we provided. So in this case, the closest hit is a ferric iron ABC transporter. So looks like this gene cluster is in good shape. Yeah, so that's basically it. So you just have 2 extra columns in this output. You have the blast hit, and then you also have the E value for that blast hit, just so you could confirm how good of a match it is to something that's in the database. MTR AB from Shuanella, no hits. Oh, there is a cystathione beta synthase. Okay, that's weird. I don't know what that is. I would double check this gene to be safe, because this doesn't sound iron related at all. But I wouldn't expect MTRA to be annotated as iron reductase in NTBI. Maybe it is. But as far as I know, there's no publicly published HMMs for MTRA. MTRC and MTRB actually got from tigrafams, but MTRA doesn't seem to be present. There's the magnetosome formation cluster in Magnetospirillum magneticum. So that's a good sign. You have cation transporters, a bunch of hypotheticals, cation. So that makes sense. So when magnetosome formation involves the formation of a particular organelle, and then inside this organelle, you have formation of magnetite crystals. So I'm not really sure how that's done exactly. I haven't looked into that pathway, but it involves ion transport into that organelle, because you need these metal ions to make them magnetite. So that's, I think that's basically it. We're coming up to the end of this tutorial. There's the output.